Hello everyone, welcome you all in this course Systematics of Angiosperm. I am Dr. Gausia Bashri, Assistant Professor from the Department of Botany, Aligarh University, Aligarh. Today we will going to study in detail a taxonomic tool, that is the taxonomic keys. Taxonomic keys are very important because all the flora and monographs are supplemented with these taxonomic keys, which are very important to reach out particular plant species via our point of interest. So first of all, we will study the taxonomic keys, what are the taxonomic keys and how to construct it, different types of taxonomic keys. So now start the today's session, that is based on the taxonomic keys. So first of all, if we define the taxonomic keys, it is an artificial device or tool or an arrangement for the determination of names of the plant included in it. So we can say that a botanical key can be defined as a device for easily identifying an unknown plant by sequence of choices between two or more statements. As you can see, there is a two or more statement. Depending upon there is a different types of taxonomic keys. And the first key was introduced in 1778 by French botanist Jean-Baptiste de Lamarck and help in the quick identification of unknown plants. This is very important because taxonomic keys save our lot of time when we have to identify particular plant from the flora or monographs. So this is a very important tool in identification for that is the taxonomic keys. And if we see a good key, a taxonomic key, a good taxonomic key is a synopsis that presenting graphically the technical characteristic which is in general or in aggregate differentiates the different taxas. So in a single pair of contradictory statement is called as couplet and each statement of couplet is termed as a lead. And if we see, the user is called upon to compare the plant under determination with the first of these pair to decide which one of the two contrasting statements appear better to the plants. So before going to the taxonomic keys, first of all we have to the description of plants. So because that's why the taxonomy has the four main branches, that is the description, identification, classification and nomenclature. Because before proceeding towards the taxonomic identification, first we have the description of that particular plant. Because of that description we cannot identify that particular plant. So description is very important, then we can go for the identification. So for when we use these particular taxonomic keys, so it is very important that we have noted down the correctly all the information of that particular taxonomic groups. So these keys are further directed to use the appropriate following the pairs until the given plant or specimen is named or identified. So we have to match our descriptions with the taxonomic keys and moving forward to one by one of by these couplets, and lastly we will reach out to the particular specimen, named specimen, that is our point of interest. Now by the use of these keys we have identified the unknown specimens to a particular named. So if we see, these are some identification keys which are necessary. So why these are the keys are necessary? As a key is essentially is a printed information that retrieval system into which one puts information regarding a specimen in hand or in form which one gets identification of the specimen to whatever level the key is designed to reach. Or in the absence of originally identified specimens of species utilized for the comparison, the published description becomes the only tool. So in larger groups, however, it is the most tedious task to compare the specimens with hundreds or thousands of published descriptions. So the task can be solved if the keys to the main groups are made available, because the main purpose of the key is to facilitate the identification or to distinguish one type of organism or object from another. So this whole process of studying the thousands of paper become very summarized due to the presence of just taxonomic keys. That which is very important because it saves our time, that is very precious, and it also give a very accurate position of that particular plants. So a key may or may not reflect ideas of evolutionary or phylogenetic system, but the construction of the keys is important job for the systematics because it identified the particular plant in very short of time, then we can proceed for the different procedures or techniques. So for if we see a characteristic of a good key, there are several good characteristics that should be present in particular key, because a key should be reliable and constant. If we are mention of one wrong entry in this particular key, then whole the flora or monograph is getting disturbed. So it is the most important criteria, that is the reliable and constant. Then it should be appropriate for the area from the plant to be identified was collected. Like I have told you, like the flora of upper Gangetic plain. So that is supplemented, if it is supplemented with the key, then it is now it has all the information of that particular area of the plants. So like for the example, if the plant collected from Delhi may or may not occur in the South India. So therefore using a key for the plant of South India, that may not help identify the plant from the Delhi. So we have to choose a proper set of keys for the identification of particular plant species. Then it can be used with ease and certainly for the naming of a plant, and it should employ macroscopic characters that are easily observable, because we cannot study at a time different all the characteristics at macroscopic level or molecular level. So keys should be present at the macroscopic level. Then the statement should be positive, and it desirable that the user is provided with a series of choices between two mutually exclusive and parallel statement called as couplet. As I have told you, each keys have a two pairs of contrasting character that is called as couplet, and then it has a leads. And the initial word of couplet should refer to a plant or plant part. And the initial word of a couplet should refer to a plant or plant part. Suppose like plant is herbaceous, either plant is tree, or stem is glabrous, or stem is hairy. So first of all we have to refer the plant part, then we have to define the character, like flower is zygomorphic or flower is actinomorphic. We cannot start the actinomorphic flower or zygomorphic flower. We have to start with the part of the plant, like the flower or leaf, like suppose leaf is ovate, leaf is acute. So first of all we have to need the mention the plant part name first. Then after the initial word of each lead, a couplet must be at the same. So suppose if we are choosing the flower, symmetry of the flower, then we have to mention the flower is first flower is actinomorphic, then the next couplet should start with the flower. And if suppose we are taking the stem character, then next couplet also start with the stem. Then the two consecutive couplets should not begin with the same word. Like if we consider the, suppose first we have taken the root, then next we cannot, next couplet we cannot start with the root, then we have to take in the some other characteristics. And suppose if one couplet uses one part of the plant, leaf, flower as a first word, then the next couplet should have a contrasting character for the identification of that particular plant. As I have told you, like the flower actinomorphic or flower, next couplet is the flower is zygomorphic. These are the two contrasting characters. Like suppose if we take the position of the ovary. So ovary, the flower may be hypogynous, flower may be perigynous, or flower may be epigynous. So we can choose from all these particular characteristics. Then both the vegetative and reproductive character are used in case of a couplet, and the initial word of couplet should be a noun referring to as plant or plant part, as I have told you earlier. This means that the key, the plant part should be mentioned and first, and the description must be mentioned afterward. Like as I have told you, leaves entire or pinnate, not entire pinnate, and the leaves like the flower actinomorphic or zygomorphic. Then the last, that the three and more character in a couplet should be avoided. So we have to choose two contrasting characters that are suited for that particular plant of group. Like just I have shown you, the flower is actinomorphic or zygomorphic, or leaf simple or compound. So these are some contrasting characters. So with this we can lead to the next couplet. So if we see a types of keys on the next, now we are going to study the types of keys. Keys are determined as device to help in identifying plant, and traditionally in flora the keys are diagnostic as I have told you, and the diagnostic character used are conspicuous and clear. So sometimes such characters are referred as a key characters, and such characters are arranged in the form of series of couplet, because every plant or every family have some contrasting or diagnostic characteristic features, and these key characters can be used in the preparation of keys, and these keys may be single access key or multi-access key. This is multi-access key is called as polyclave. And if we see the single access key, which is also known as the sequential key, analytical key or pathway key, and almost all the single access keys are dichotomous, that is dividing into two pairs of keys, and it consists of opposing statements that each statement leading to the another pair of choices. This is called as the couplet, and consecutively narrowed the user search down and until a final statement identified the organism. Now, so with the help of these keys, we identified an unknown specimen. And these different kinds of keys are used in taxonomic work, and most important one is the listed below, like the single access key, in which two most important are the bracketed key and intended key, and there are several types which we will discuss later. So the most common two types of the single access key are the bracketed key and intended key, and the first one, that is the bracketed key. In a bracketed key, each couplet has its two leads immediately adjacent and one below the other under the same number on the left hand side of the page. And the first couplet to be read stand at top of the key under the number 1 on the left hand side of the page, and opposite the lead on the right hand side of the page. Subsequently, number 2, 3 are used as a reference number further down the key. In a bracketed key, it is by use of the number that is determiner is directed to the next appropriate couplet to the console. This will clear in the next page if we see the bracket key. Suppose we have to identify the different groups of plant according to Bentham and Hooker. As we know, Bentham and Hooker classified the plant into main three classes: dicotyledony, monocotyledony and gymnospermae. And dicotyledony further classified into three subclasses: polypetally, gymopetally and monoclinidae. So suppose we have to identify these group of plants. So first of all, as you can see here, this one showing the plant with seeds and ovules enclosed within ovary. So we all know that only angiosperm plants have a ovule enclosed within a ovary. So now this will lead to the second number, and the second couplet in the first one has the plant with seeds and ovules are naked. So as we know, the gymnospermous plants have a ovule that are naked, they do not have a enclosed with ovary. So here we have identified the one group of plant, that is the gymnosperms, because they have a naked ovules. Then from the first one, as we have moved to the second couplets, the plant with seeds and the ovules enclosed within ovary. As we know, these are the angiospermic plants, and if we have to characterize further them, then the flower of mostly tetra or pentamerous. As we all know, the plants which have a tetra or pentamerous, generally they are belong to the dicotyledony, and embryos with two cotyledon. That means it is representing the dicotyledony, and for this we have to move down to 3 and 4th number. So the next couplet in this, the flower mostly trimerous and the embryos with one cotyledon. So we can see this group of plant are also called as the monocotyledony. So if we see the petals and sepal both present, then we have to move to the 4th number. Then petals or sepals are rarely or none present. We know that the Bentham Hooker classified the dicotyledony in three groups, in which polypetally have a free petals, gymopetally have a fused petal and monoclinidae when there is no distinction between the petals and sepals. So from here we can see then this petals and sepals, if you both present, then we have to see the number 4th couplet. Then if we 3 has the petals or sepals rarely or none present, then it is a monoclinidae. Similarly, petals if free, then it belong to the polypetally, and petals are fused, that belong to the gymopetally. So you can see here from this simple bracketed key, we can identify the gymnosperm, monocots, dicot, and dicots. We can identify the subclasses monoclinidae, polypetally and gymopetally very easily. So it is just a simple type of a dichotomous key that is called as the bracketed key. So, if we see the advantage of bracketed key, when it properly constructed, one can quickly and easily run through this key both forward and backward, and it is also more economically in terms of space because it is unintended. And the disadvantage of this particular is the both simple and bracket key is the relationship of division is not apparent to the eye like in case of a intended key. The next one is the in single access key is the indented key. As the name indicates, it is also called as the yoke key. In this type of key, each couplet has its own leads indented by the same amount of the left hand edge of the page. So, the first couplet of the consulted is one of the least indented and the has first lead at the head of the key, and the next appropriate couplet that is to be consulted has its first lead placed immediately below the chosen lead to the previous couplet and so on. So, an intended key is sometimes provided with accessory numbers to help the user specifically if the key is too long. So, such number help the user to easily pick up corresponding couplets. As you can see in this one is the similarly the intended key as we have seen earlier. As you can see here, the plants with seeds and ovules are enclosed. As we know, these are the angiosperms. Then, as you can see, further the second number showing the flowers are tetrapentamerous, and then calyx and corolla both present. Then, if petals are free, we can say it is a polypetally, and petals are few, then we can say it is a gymopetally. And this the contrasting pair of this three numbers, as you say, calyx and corolla present or both option. As you can know, the monoclamidae has they don't have a distinct calyx and corolla, they are termed have a perianth. Then if we see the flower here tetrapentamerous, then flower here is the trimerous, that means they have a one cotyledon and they belong to the monocotyledon. And just the flower plants with seeds with the ovule enclosed, and here you can see the flower plants with seed ovule naked that belong to the gymnospermae. This is the typical example of a intended key. And if we see the advantage of intended key, this type of key is advantageous in the sense that the relationship of various division is quite apparent to the eye and can be used in reverse also. And it is good for purpose to key to higher text or comparative keys. And the disadvantage of this particular key is when the key is short, it works alright, but in long keys, the alternatives get widely separated and take more space. So, moreover, this can lead to the much white space in the print and consequently little remaining room for the lead text and illustrations. So, there are some advantages and disadvantages of intended keys. The next one is the multi-access key or polyclaves. And this is also a dichotomous keys mentioned earlier are based on the principles of one entry point, that is single access, sequential access. But in order to improve the efficacy with which the determination of the name of taxon, that is the multi-access or multi-entry order free keys have been introduced. These are also referred as the polyclaves, and such keys are user oriented and can be commenced at any position. Unlike the dichotomous key where the author provides the characters, in multi-access key the user select the characters and not the authors for the identification. So, also unlike the rigid given order of the choice of character to be used in single access key, the user in a multi-access key can choose not only any character but also in any sequence. So, it can proceed even if the information about a few characters is insufficient or unavailable. So, it is a very important in multi-access key is usually produced in the form of punch card, and this punch card, that is the by Bianchi and Clark first used punch card, and the Clark prepared such multi-entry performed keys for the identification of hardwoods, and there exist two kinds of perforated or punch keys. The first one is the H punched key. As you can see here, multi-access key, the first one is the H punched key. In this kind of punch key, a given card represents a taxon, and each card has a number of character or attributes, and each attribute is represented by the hole of the margin of the cards. As you can see, these are the different types of holes. So, this one is a figure that showing the H punch card, and it is given taxon possesses that character, the given hole is notched. So, the card of different taxa are then stacked. Then the taxon cards are sorted by the use of large needle, and the user runs the needle through the characters hole of card, that is the say like leaves are opposite. So from all the stacked cards, one of the needle is pierced through the leaves opposite, and the holes only the cards notch for the character that leaf opposite shall fall down. So interestingly, the choice and the order of selection of the characters are solely decided by the user. Then the fallen cards are stacked again and another character is chosen where the needle is to be pierced. Then the process continues, only one remain that represent the unknown taxon in this particular H punch keys. In this image you can see the H punch card of the genus Ranunculus. Here only the attributes represent in the example above as pictures, and many more attributes could be added along the vacant hole to make the identification process more versatile. And the next one is the body punch card, which we can see in the next slides. The next, that is the body punch card. In this kind of key, the holes are punched in a row on the main body of the card, unlike the H punch card where each card represent a taxon. So in body punch key, each card represent a characteristic, that is the attribute, and the numbers are printed on the card to point out the standard position of each taxon. So if the taxon possesses that particular attribute that mentioned in the card is positioned in punched out, then the cards are stacked and held against the light. When only one hole shows through the light, that taxon represents the identity of that unknown plant. So in both the above two kinds of card key are operated by user in step-by-step elimination process. So however, any character and in any order may be used. So such key can be used as a partial key, which is very nowadays popular. And if you see, a multi-access key contain more information than the traditional dichotomous key, because these dichotomous key is prepared by some other author. The present author does not have our own on this particular, so it contains data necessary to construct many different dichotomous keys. Thus the term polyclave is used for this type of keys. And with the help of computer software, it is possible to convert multi-access key to a computer interactive format. And nowadays, computer assisted animation process such as Delta, that is the descriptive language of taxonomy, or Lucid system are important tools for future means of the plant identification, which is a very important. So beside these, there are some other type of keys are also exist, like the pictorial keys for the field workers and non-taxonomists that can identify the commonly occurring species with the help of the characters together with their figures in a comparative manner. So there are several such keys for the various groups of animals especially. Then the box type keys, like the branching and circular key, the box type key is also meant for the field workers and quite useful for the quick identification of most species. Like you can see, this is the identification of 5 kingdom system, like the Monera, which don't have a nucleus, then Protista, which has a nucleus, then Plantae, which are autotrophic, then Animalia and fungi. Animalia is mobile and the fungi is immobile, and these are the heterotrophic, and these are the only one cell organism, and these are the multicellular organism. So we can identify these 5 group of plant and these 5 groups of organisms by this box type of key. Similarly, there is a next is the circular type key. This key is also meant for the small group. It is also quite useful for the non-specialists, especially field worker who want a quick and immediate identification of commonly occurring important species. This is also, you can see here, this is the circular type key for the 5 kingdom organisms. As you can see, this one is the organism with one cell, this one is showing the organism with more than one cell. Then has nucleus that belong to the protist, no nucleus that belong to the Monera, autotrophic, that is the plant, then heterotrophic, maybe mobile, that is the animal, immobile, that is the fungi. Then branching type keys, as I have shown you in the keys section in the taxonomic aids, such keys are also meant for the easy and quick separation of the species as far as the group of is small. So it is quite useful for the field worker also. As you can see, this is the organism with one cell, that is the no nucleus with, that is called as Monera. Then those who have a nucleus, that is called as Protista. Then organism with more than one cell, may be autotrophic or maybe heterotrophic. The autotrophic that belong called as plants, then heterotrophic may be mobile or immobile, and the mobile one is Animalia, and the next one is the fungi. Then the next is the computer aided key. As we have seen that the multi-access key is now very important, and in recent years the computer technology has been used for the constructing keys. So figures, photographs can be fed into the computer with the help of appropriate program, and it is much easier and less time consuming to compare the characters of unknown taxa with known taxa with the help of these computers. So scanners of computer can observe and record the characters of undetermined taxa and compare this with those of already known plant groups. And in this, the delta, that is the description language for taxonomy, is a useful system for programming for recording taxonomic descriptions of processing by computer and generate conventional identification keys to identify the group of plants. Then in modern technology has provided a digital platform for the identification, as databases for plant of different parts of the world are now available to help in identification. For example, the flowers of India, that is had the website eFlora of India, and recently these have introduced a QR code on the page of different species, and this can be used for obtaining details about the plant. So similarly, the Center of Ecological Studies, Indian Institute of Science, Bangalore launched an online database for the plant peninsula India on March 2009. So we can easily identify a particular group of plant from just scanning its QR code and match them in online system. For the proper use of these keys, we have to follow some certain rules, like appropriate keys to be selected from the floras, manuals, handbook and monograph, etc. And we have to introductory comments to be properly read first. Then both leads should be read before making a choice. And the meaning of unknown terms should be checked using the glossary. When the measurements are used, such as the length or leaf, fruit, etc., several material species to be measured, and no decision to be taken based on a single observation. Then result should be verified by reading a description, comparing the specimen with illustration on aesthetic herbarium specimens. Then there is some merit of these keys that facilitate the identification of a specimen. Keys are dichotomous, offering two alternatives at any point, easy to apply, and provide best assurance for correct identification. It provides tools for taxonomic analysis, since in the preparation of key, one must select, evaluate and arrange taxonomic characters. And the keys character apply usually to all the individuals of the population regardless of age and size. Besides this, it also has some demerits, like construction of keys are time consuming task. Key does not provide biological and phylogenetic characters. It does not provide the information of cytological and molecular characters of a specimen, and it is not useful in case of identification of all the specimens. So these are some demerits of this taxonomic keys. So that's all for the today. But this taxonomic tool is very important in case of a practical classes at the graduation or PG level. So that's all for today. Thank you all of you for joining this session. Thank you all.